Hi, my name is Rachel Thomas, and I'm going to be speaking about using machine learning to identify the lab of origin for genetic sequences. So this is based on the paper, A Machine Learning Toolkit for Genetic Engineering Attribution to Facilitate Biosecurity, published in Nature Communications in December 2020. And this was published at the same time as an accompanying perspective paper on the biosecurity benefits of genetic engineering attribution. The key question the researchers asked is, given a bacterial plasmid, can we accurately identify which lab genetically engineered it out of over 1,300 labs? The goal is to create an attribution toolkit to promote responsibility and biosecurity. And the authors are clear that in a forensic investigation, this should just be one tool of many used. It is not intended as a standalone. I think this is an interesting problem. It's inherently multidisciplinary. It draws on machine learning, genetics, and policy. And this research paper is focused on the machine learning and genetics aspects, and the accompanying perspective paper I mentioned gets more into some of the policy implications. So the data source for this project was AdGene, which is a nonprofit online database of plasmids. So the model, it's kind of shown along the bottom row. This is figure 1A from the paper. It was created with this data from AdGene. And then once, if the model were to be deployed, if a new bacterium was discovered, lab forensics would be run, the DNA would be sequenced, as well as phenotypic properties could be tested for the bacteria. And that data could be put into the machine learning algorithm, and the output would be a set of the top 10 most likely labs with accompanying probabilities of what the lab of origin is. The inputs to the model would be the DNA sequence, and then the phenotypic data includes resistance to various antibiotics, the number of plasmids per cell, the strain used to clone a plasmid, the growth temperature, and so on. And the authors did two different versions of the model, one where they used all this data and one where they just used the DNA sequence. Figure 1 kind of gives an overview of this project. Figure 1b shows how they encode the input data. And this is interesting to me. They're using similar techniques to what's used in natural language processing of just English language or other languages as well, but of kind of how people speak, but using this for DNA sequences. But basically they were finding the most common subsequences and replacing those with tokens. So here ATG shows up a lot. That's being replaced with Y. And this both compresses the data, so it takes up less space, and it also is capturing some underlying patterns before they've even put it into their model. In figure 1c, they show that the learned motifs of kind of what these common subsequences that show up are biologically relevant, and the longer high-ranked motifs include fragments from promoters and plasmid origins of replication. And that's a nice feature of this model, is that it has this biological relevance. In figure 1d, they show so the next step is to take these sequences that have been tokenized and now embed them and enter them into a recurrent neural network. And in fact, the name of this model is deterrent, spelled with an RNN, which stands for recurrent neural network. It's a bit of a pun. And this is a common and very effective type of model for language for sequences, and something like this that's in variable length. And then the output, as I mentioned before, you can see it kind of on the right of 1D, is these probabilities for the lab of origin. In figures 1E and F, they're showing the accuracy. So K equals 1 is how accurate is it at making the top number 1 prediction. This is the lab of origin. And then in red, K equals 10, that's for getting the right lab in their top 10. So of the top 10 labs predicted, was the lab of origin, the true lab of origin, one of those. So on the far left is deterrent, the model that includes both the DNA sequence and the phenotypic data. 1 over is that model just with the DNA sequence and without the phenotypic data. They also compared that to BLAST. They compared it to a different type of neural network that's more commonly used for image data. They compared it to a baseline where they just guessed the most common labs. And then they compared it to uniform, which is just randomly picking one of the 1,300 labs, and that was so wrong that you can't even see an accuracy bar. It was 0. And then for figure 1F, they've zoomed in on their 2 versions of the model, the DNA sequence with and without phenotypic data. So a nice attribute of this model they've created is that they can calibrate it for confidence. And so machine learning models, it's possible, in fact, many high-scoring models are accurate, but also very overconfident. And so here they have a temperature parameter that's shown in 2A and 2B, and it's colored in. But basically this parameter is used to calibrate the model, and it's just adjusting its confidence. And so figure 2C shows the model before they calibrate it, and it's overconfident. So they have indicated accuracy with a dash and confidence with a dot, and the dots are almost all above the dashes. And then figure 2D shows the model after they have used the temperature parameter to adjust it, and now the dashes and dots are much closer together. So that previous section I talked about was focused on predicting the lab of origin. The next problem they tackled was building a different model to predict the nation of origin. And this could be useful if the bacteria originated in a lab that doesn't have any existing data. Can you at least identify which nation it's from? This they divided into 2 subproblems. And so the first is just predicting whether it is of domestic or international origin. And here they're using a much simpler model. They're no longer using a neural network. They're now using a random forest. And so for this problem of domestic versus international, BLAST actually does slightly better than their random forest, although they're quite similar, and that's what figure 3A is showing, and then 3B is just the area under the curve for their model. From there, they move on to if they determine that a plasmid originated from an international lab, now predicting the country. And so this is a multi-class prediction problem. And they've created a random forest for this, and now they are outperforming BLAST. Again, they divide this into measuring the accuracy on getting, the #1 correct choice versus getting is the correct one within the top 10. And again, since this should be used as a tool of many in an investigation, this could be useful in narrowing down what are the top 10 potential countries. This could have originated in. Figure 3D shows a map of how the accuracy varies by country, and figure E shows that this is somewhat dependent on the number of training examples, not surprisingly. So countries where very little data has been put into the AdGene database, the model is not very accurate at predicting them, whereas countries that have shared a lot more data, there's higher accuracy in predicting them. Next, they tackle a slightly different angle, which is just looking at collaborations between labs or dependency of work. Sorry, I should say ancestry dependence. And so this can take the form of collaborations or published work that builds on one another. And so figure 4A is just kind of these trees showing as a plasmid or the work of a lab on top, and then how many kind of branches coming out of it, that's the work that depends on it. Figure 4B shows the distribution just of the number of plasmids per lab and the number of plasmids per country. And then that can be compared to figure 4C, which you should note, so this is on a log scale, these are both on log scales. This is extremely skewed. So basically, and this is capturing dependencies, there are some labs, or a very small number of labs and plasmids that have, in the case for the plasmids, thousands of dependencies. In the case of the labs, hundreds of dependencies, and then that quickly drops off. So you can see kind of this really steep drop off here and here, with most labs having, this looks like it's at 0, no collaborations, and most plasmids having 0 dependencies. So that's considered a power law distribution. In figure 5a, they're showing, they're kind of using machine learning to cluster these plasmids and showing that the probability of being accurate is highest. So this is color coded. The dark blue ones are the plasmids that are kind of farthest away from the others. So if a group of plasmids is distinctive and it clusters away from other plasmids, then there is a higher predictive accuracy on those, which makes sense. I like the experiment that they do in 5B and C, which is that they took two plasmids that weren't in their data set. So they, for 5B, they took one that was in GenBank but not AdGene. And they show that, so the plasmid in 5B, they know is from Chris Voigt's lab. And they know that Chris Voigt does overlapping work or work in the same area as Baojun Wang. And they show how the algorithm basically predicts those two labs with approximately equal probability. And so that kind of fits with what you would expect. They're doing similar work. And then for 5C, they took a plasmid from the Edward Boyden lab that was not included in their data set, and they deleted some of the restriction sites in it. And restriction sites can often be a hallmark for a lab group, and they showed how this really decreased the probability to kind of delete these signature restriction sites. Figure 5E, they custom made a plasmid combining the work of two different labs. So they took plasmids basically from Ed Boyden and Omar Akbari's labs, two separate labs, combined aspects of both, and showed the probabilities. And here, this is kind of you can imagine it sliding a window over the whole plasmid and giving the prediction at different points. And overall, algorithm gave a higher probability to Omar Akbari, but there were points where it thought it was most likely from Ed Boyden. So in conclusion, the benefits of this model is that it achieved 70% accuracy on the lab of origin problem. The previous best was 50%, so this is a big improvement. It's a motif-based sequence model that has biological meaning, and so that's nice that that was kind of captured in the model, and it takes advantage of phenotypic data. The calibration, which gives a confidence score, can allow this to be weighed alongside other evidence in a forensic investigation. And the authors argue that this could increase openness and attribution because it can be used to find ancestry dependence of kind of work that depends on other work. That might encourage people to be more upfront about that. And the authors note that future work is needed to build larger, more diverse data sets, since the data used here, over 50% of it was from the US. A few limitations I saw with this work. I worry that it could disincentivize openly sharing work, just in that I worry that countries could be overly cautious and kind of not wanting to share work that they feel like could later be used to incriminate them, rightly or wrongly. It's always important to keep in mind that a machine learning model is only as good as the data used to train it. And so if a bioweapon was created under different conditions with a different team, different design principles, it would be unlikely to match patterns in existing data. I should also note that this paper did not consider an adversarial approach, which I mean, that would be very hard to test of if someone was trying to keep a body of work secret and not including that in the data set. They could potentially, also make sure that kind of related work is not added to the data set. It's also important to think about the harm of an incorrect prediction. This could have huge geopolitical implications. And also to think about that combined with automation bias. And so automation bias has been documented in dozens of studies, but it's the fact that people tend to trust computers and algorithms to be very accurate and to not make mistakes. And so that could have particularly damaging implications if one country identifies or predicts that another country has released a biological weapon. Also, this work was limited to bacterial plasmids. It doesn't account for viruses or non-plasmid bacterial sequences. And here is a list of the references I consulted. Thank you.